I'd like to thank Margot and Karen and Judy and all the other people, Günther, for inviting me. And it's a great pleasure to be here. I have never been speaking to an audience with so many women, so it's wonderful. And so here I'll report on a project which has been going on for 2 or 3 years with a bunch of collaborators and co-lead Irvin Fry, a biology from Lawrence Berkeley lab. I just want to advocate for what we call now a holistic approach to data science, which is not just one particular domain, but it's really everything integrated. So a lot of things I speak, I think, can be mapped to the business world, like the domain science. Like I work on remote sensing, neuroscience, genomics, and also embedding there is the personal skill to pull everything together, and that's kind of related to the art part. And also you need the more hard muscle type of analysis, analytical, but also the view on theoretical statistics now I take is really you have multiple idealized models to analyze something already proven practice empirically. And then you definitely have the technology software to support. So domain knowledge in business like what in the last talk, like a merchandise. And in terms of art of the data science, I recently in an internet blog rebranded applied statistics to data wisdom. So it's very much copycatting at the time, the wisdom definition in Wikipedia actually has changed. So I couldn't find it. Right now, somebody edited it, so it's not the same. So I would say that data wisdom is the ability to combine domain, mathematical, and methodological knowledge with experience, understanding, common sense, insight, and good judgment in order to think critically about data and to make decisions based on data. So it's kind of covered the self side of interpreting, connecting with reality. So there are set tens of questions to help people to gain I would call data wisdom ability, basically from teaching a PhD level course in statistics, applied statistics in Berkeley for the 7 years. So the topic of today is really about spatial genomics data. Jennifer, in her first talk, alluded to some genetic problem. So here she talked about protein-protein interactions. And what I bring in is we're going to end up with a network, but the information here is spatial. So it's kind of like co-occurrence or association by guilt. So when you know 2 genes are expressed and co-vary in certain regions of location, then you conjecture they probably interact because they seem to go things together. And what I'm looking at is really part of system biology and this Berkeley Drosophila genome project. So Drosophila actually share about 75% genes as humans. So it's very cheap and a model organism to study a lot of gene-gene interaction, a lot more advanced than yeast, but still not as advanced as us. And we're looking at, in the first 1.5 hours to 3 hours development to understand how the fruit fly embryo build different organs. So think about building a house. They want to build different rooms in the house. And each gene you think of as a worker. And the worker will collaborate with other workers depending on what they are building. So that's kind of a good metaphor. And from traditional biology, we know that they compartmentalize the building process, like these different regions, like different rooms in a house. And we use this fit map, and with 7,000 genes with 1 terabyte data in the project, try to reconstruct this kind of schematic map as a first validation. I hope we'll do better than the old technology. And this is a proof of concept to show that with something called laser ablation, a woman scientist from Germany, she's actually did this for very young, Christine Newson-Wanhart, got Nobel Prize in 1995 for her work in the 70s to figure out this gap gene network. This gap gene network is really like setting up the Y coordinate system, like the division on the stripes for the Drosophila embryo. And lower there is the human embryo. You can see that we're not that different from a fruit fly in the early stage. We segment our body into different parts. And if you look at the net gene, so this oval is really like embryo. And you see these 2 orange color HP, that means hunchback. So if you knock out that gene, the embryo will become like a hunchback. And tailless means you knock out that gene, it won't have tail. So it's very descriptive. And giant means become giant, big. So you see 2 blues and 2 orange. It means that at the anterior part of the embryo, giant interact with HB and KR, and in the posterior will interact with another bunch of genes. So basically, this worker, giant, will work with different teams to do different things. So this is already known. We're going to use this to validate what we find. And this is kind of our overview. Oh, how do I go back here? Of our framework. So we have a team of statisticians from my group, a team of biologists from IBL, and a team of computer science from Tsinghua University. And the whole framework is like we do the statistical machine learning part, and we go to wet lab to do knockout experiment, and then the Tsinghua team will build up open source software on Spark and Fiji, which is biomedical open source, to for other people use similar data, can use our development to do their science. So it's a whole, basically 3 subjects coming together. And right now, we are using actually something sparse coding or non-matrix matrix factorization. Later, we want to probably deal with small hypothesis testing and data perturbation. On the CI side, they have to deal with data metadata management, data-driven automation, and open source. So we kind of bring it together through this we call CISPA framework. And so while I was brought down to the project, then I was working on computational neuroscience. Too bad Feifei left. I was going to thank her that we actually used ImageNet she spoke about, and we were writing a paper called Artificial Neurons Meet Real Neurons. So what we do is that we use deep learning and take the last layer out with the artificial neuron, and we have a single neuron for we form a cake. So we put it together with the last layer trained use real physiology data, but the first few layers use artificial neurons, and we do really state-of-the-art prediction. And so at the time, I was still working on computational neuroscience. When we have images, one thing come to mind is, can we do this decomposition, which happened very nicely with computational neuroscience? You have these edge detectors. Indeed, we can do that. We later moved to non-negative matrix factorization instead of exactly sparse coding. So we just put our images into, there's tons of work which I didn't speak about. 80% work is about processing, so we can do that. It's a huge amount of work, registration, denoising, all kind of things before we can put that into the same grade and like a matrix. Somebody talked about a bulldozer and become a matrix, and that was it. Lots of work. So each image become a vector, and we didn't code the spatial relationship. So just you can scramble the rows, it's fine. And then we did a non-negative matrix factorization using a package called SPAN by former postdoc Julian Morel. And I don't have time to give the details. And we developed some stability-driven criteria to decide how many components, and that's what we got. So those are the 21 we call principal patterns. And when we showed it to biologists, they really loved it. They see meaningful patterns. You see the stripes? It's almost like vertical and horizontal coordinate system. And you have anterior and posterior about the brain and the guts. And then you can lay it out. Oh, this is not projecting very well. So the colors are a little messed up. So you can see that we can map qualitatively to this feed map, which is not precise, to say that the red is like about the gut, and there's foregut, there's about skin, there's about the stripes, and they are biological meaningful. So that's just visual inspection. But then you have to go further than that to validate why this decomposition is useful. And what we did is also to see that use this to characterize all the genes. So we did another post-processing using lasso to make things even more sparse and say that, so different genes, each pattern you can think of as a different room in this building up of different rooms. Okay, so I have 21 rooms, and we look at which genes have substantial expression in that room, and we said that gene is working in that. And then you look at all the genes. Here I plotted like if 2 rooms are close, and I plot how many genes they share. Those are proportion on the vertical axis. So you would think that the 2 rooms are very close, the principal patterns are very close, they should share a lot of workers because it's more efficient. But then you see the whole pattern kind of going back. So that means there are rooms quite far apart. One is the anterior, one is the posterior. They share a lot of genes too. And you look at them, it turns out that they are the foregut and hindgut crosstalk because they are the part of the digestive system. They share the same tissue. So of course, they share similar genes. So it's not just distance, but also the functionality of the rooms also share the same workers, right? You have a kitchen, you have a bathroom, they share a lot of same plumbers. Similar idea here. So it's just something to validate that what we get is meaningful. And then we went back to that gap gene network. That cross is also off. So that should be a little high up between the knee and joint in the back. So that's got off too from the reprojection. So for the 12 links in this gap gene network, we were able to reproduce 11. So that was not bad. And the other one, we got the opposite sign. We think it's because when we had the embryos manually labeled to this particular early stage, 4 to 6, there's mismatch because it's not precise. So things not simultaneously happening, and therefore they look overlap, but in reality, if you have simultaneous measurement, they actually don't interact. We think that's the reason. But Church's group from Harvard last year had a paper saying that they can actually simultaneously measure 1,000 different gene expressions. And if that become reality, that's really a good application of what we develop here. We now have mismatch over time because it's humanly visually inspecting, say, at the kind of early stage. So the timestamp is very coarse here. And then the next is like, this is suggestive. We see links. And one thing I didn't point out, you couldn't see in that network for that particular room, that's we think how they interact. There's something called CG, I don't think you can see. CG means computational gene. That means they know their genes for looking at the binding and the product, but they don't know what these genes do. And some of these CG genes in our data show very strong connection to this gap gene network. And I've been told it's huge. You find a new member of the gap gene network because gap gene is supposed to, people think it's complete. And it's such a famous gene network. So that's what we did. So we took these genes, 4 CG genes, and we took 3 gap gene networks and find the strand links. So the design criteria at this stage, it was qualitative, was that we want to find new members of the gap gene because it's famous. We also want to discover things people haven't seen before. So it's both confirm things we know or close to something we know and new discoveries. And then last year, actually almost more than a year ago, we started doing, the lab started doing knockout experiments. And that took a long time. So we just finished the first paper without the lab result because I've been waiting for the lab result to come up, but we decided that we just cannot wait anymore. We have to publish the more predictive and the decomposition part. So the technique, why it took so long was because the lab, Sue Seneca's lab, for the first time used CRISPR, Cas9 technology to do the knockout. So CRISPR Cas9 is attributed to 3 people. Jennifer Doudna, actually in Berkeley, Emil Charpentier, I think now in Germany, and Feng Zhao in Broad Institute, and Berkeley and MIT in a patent fight about who owns the CRISPR Cas9 patent. And somebody said they should fight forever, and therefore everybody can still use it. So some people don't want them to resolve. So it's basically a molecular scissor, which borrowed from the bacterial defense system against viral invasion. It's like a scissor. You can cut very precisely, relatively easily, where you want to cut. But actually, there are different versions of it, as I learned. So when you say use CRISPR, depends on which base pair you choose. Actually, you don't get the same result. So knockout is not really single. I'm not worried about that right now. And here are some results. So for the first line, they are so what being knocked out was CG13894. And the first row are the wild type normal embryos. And then the lower panel has the mutant images. And for the middle one, we're looking more closely, the Fushi Terazu, definitely we got something because you knock out that gene later, not at this stage, the embryo actually die. So definitely it's important. But we're not too happy because that's not, we want something more precise and quantitative than it's important. So we think our prediction is that we'll actually change the gap from our data. It seems like it's interact with a gap gene, and we think the spacing will change. But that turned out to be very difficult to see because there's variability within different embryos, and how do you measure the stripes? So we're in the deep investigation of how to make sense of these randomized knockout experiments. And that has been going on for a month. And we have a lot of slides of embryos which haven't been looked at. So it's become very, very labor intensive. And we might have to look at higher precision data. So you can see that humanly might be something, but definitely we know that gene kills the fly, but that's not the most impressive quantitative prediction. So we hope to get more. So that's where we are. And of course, we try to provide a platform for other type of genome data. And for this RNA-seq data, you don't have spatial information, but you have different conditions, kind of a surrogate for space. And we tried it very briefly on multi-encode data, which the lab also produced. We do see similar reasonable principal patterns, right? But we haven't investigated enough. And as I said, this cloud-based infrastructure for system biology analytics based on Spark and Fiji hopefully can be used by other labs to do their own decomposition. I really see this sparse coding and negative matrix composition as a way kind of modern PCA when you have high dimensionality with enough constraints. It's decomposition, and it's a linear fashion, and has been empirically proved useful in space sparse coding, computer neuroscience with physiology support. So I think that's what would be useful for other labs as well. And here's like what we hope. We hope to develop new statistical machine learning methods, but very validated by this particular scientific investigation, and also have computer science development and understand gene-gene interactions. And eventually, we hope to provide insight, also suggestion for human genetics, because we share 70% of the genes. And this is really a data science done by multidisciplinary and international team. And that's a shameless plug for my last year's presidential address, which traces history of data science back to 1890 census. That's where I think computer science, the first tabulation machine called Herowith, was developed because we have to do census in 10 years. Here's the team, and I was saying that we have a fused team brain that you basically need the interpersonal and leadership skills so we can talk and really tap into each other's brain with the knowledge we need to solve this problem. And my student, Siqi Wu, has been really the great team member and do a lot of the work on that. And my co-lead, Irvin Fries, in Sue Seneca's lab. And actually, my postdoc, Anthony Joseph, now is warm my lab. So you can see the training on genomics is useful probably for industry as well. And the team at Andy Yao's Institute in Tsinghua, led by Wei, and the sponsored agencies. Thank you.
Thanks very much.